Okay, very abridged intro. Thanks, Foster, for coming. Foster is a grad student in Amy Keating's group. I'm going to give him another introduction after he ends, just so we round out the whole talk with yet another intro for him. Thanks, Foster, for coming, and we're looking forward to your talk.
Great. Well, yeah, thanks, Nick, for the invitation. And I'm happy to talk today about improving protein binder design by learning sequence structure compatibility. And as a brief roadmap, I'm going to begin by going over this, a background before diving into the methods and then the performance. So one way to look at computational binder design is as a sort of series of generative and scoring steps, where let's say you have some system that you're working with, you want to design a new binder that's going to modulate something in that system. You probably have a lot of targets that you could potentially design a binder to. So right off the bat, there's sort of like a scoring step, a filtering step, where you need to decide what targets you actually want to sort of progress with. You'll then get a couple of targets. You'll do some lead discovery, which you can do computationally nowadays with tools like the Fusion tool, like RF diffusion, or other computational tools. You'll generate tens of thousands, hundreds of thousands, potentially of designs, but we can't experimentally test all of that in the preclinic or clinic. So we need some way to do lead optimization, and I work on like computational ways to do this. Computational ways to take a set of possible designs and score them according to some likelihood of actually accomplishing the desired binding effect. So what that really looks like is we have some target, we have designs, a lot on peptides, which is why the designs look like they do, very sort of flexible. We have their structures and their sequences. And the goal of a scoring algorithm is to take in these structures and sequences and assign some structure sequence compatibility scores that represent how likely the binding event is to occur, with the goal being establishing some sort of ranking, some sort of scoring that you could then use to prioritize what designs you want to pursue. The computational scores generally have an efficiency effectiveness trade-off, where you could have a model that's very simple, something like for small molecules, Lipinski's rule of 5. You can apply it to as big a library as you would want, and it is pretty effective itself, but it's not as effective as maybe applying some energy-based model that you have developed specifically for your system. And what machine learning has done over the past couple of years is it's taken this efficiency effectiveness trade-off for computational scoring models and just sort of moved the benchmark. It's pushed everything further to the right. So now we have models that are both more efficient and more effective, but crucially, they still exist on this spectrum. So you have models like AlphaFold Multimer. We maybe want to use AlphaFold 3. Well, you can only predict structures, like 20 structures per day until they release the full code in November, right? So AlphaFold 3 it's great. It's a great model, super effective, but at least right now, pretty low throughput. You have other versions of AlphaFold that have been developed since it was released a couple of years ago. One I'm going to be talking a lot about later in this talk is AlphaFold Initial Guess, which was developed by David Baker's lab. And it was developed sort of specifically for this purpose of scoring possible designs. And it works without multiple sequence alignment for the binder, so it's a bit faster than the sort of base version of AlphaFold. And again, works specifically for this binder scoring task. And then you have other methods like ESM-fold, Omega-fold, which are even faster, but not quite as efficient. Now, I'd love to be able to stand up here today and say that what I've done, what my work has done, has pushed us even sort of further on this efficiency effectiveness sort of landscape. And there's some evidence that my model is like both more efficient and more effective than the state of the art. But what we've really done mainly is make something that is more efficient. So that's sort of the space that we're occupying. Is we've built a method, a model that is extremely efficient, and for a couple of use cases, which I still think are really important, is also more effective. Okay, so that's the background done. Let's now dive into how we've actually done that. So we looked at this problem of improving structure sequence compatibility scoring for proteins. And we used an analogy of looking at image and text compatibility scoring by machine learning methods in natural language processing and computer vision. And this is a big problem that field has been working on. And in 2021, OpenAI, which as you might know from their GPT models and DALL-E models, they were also working. They were working on this problem. And the problem is basically you have an image and you have a set of possible captions that you want to be able to map to the image. And you want your model to be able to score the different possible captions. And you want one that can identify that this picture is a picture of guacamole instead of a picture of any other type of food. Okay, so how did OpenAI go about solving this compatibility scoring problem? Well, they developed a method called contrastive language image pre-training, or CLIP. And what they did really is they recognized that by the point where they were developing this in 2021, there were extremely good architectures for encoding text and images. They developed like GPT-2 and GPT-3 by that point. There have been image encoders that were trained on millions of images already that could develop very informative representations of a bunch of text that you want to provide to the model and a bunch of images that you want to provide to the model. But although these sort of separate models were very informative, they produced sort of computational representations of this underlying data that were informative, they couldn't be directly compared. And what I mean by that, sort of maybe more clear once I go through this example. Let's say we have some text about captions of dogs, and then we have some images of the dogs. And let's say we take our text and we pass the text through a text encoder. Now, in reality, that's going to generate some extremely high dimensional vector that exists in this high dimensional latent space. But for our purposes, I'm going to simplify things down to just R2, so Cartesian coordinates. And because these text encoders work, they will encode similar text, like similar semantic texts, to a sort of related space in the latent space, because it knows that it can identify that there's a dog in the text. So maybe those dog-related text encodings will end up somewhere in this latent space. And we can take images and we can encode them as well, and we can map them into the same overall space, the same dimensionality space, and we could see where the image representations live relative to the text representations, the text encodings. And the dog-related image encodings, because this latent space is sort of arbitrarily designed to work with text and images, I mean, all it is just the shared dimensionality, there's going to be no inherent relationship between an image encoding and a text encoding, even though both data modalities are fundamentally about dogs. So in other words, the cosine similarity between a random image encoding of a dog and a text encoding of a dog, that will be low if we don't fix this problem, right? And whereas ideally, especially if we want to do the scoring sort of task, we would like the cosine similarity to be high, because then we could just use the cosine similarity as our sequence image compatibility score. All right, so that's the objective. So how did they go about doing that? Well, that's this contrastive learning process. And the core idea is that in CLIP, they form mini batches of text and images. And during training, the objective is to maximize the cosine similarity of true pairs of text and images. That's like the diagonal of that box on the left here. They want to maximize these cosine similarities, and they want to minimize the cosine similarities off the diagonal. And I'm going to talk in the next slide about how they actually do that and some of the sort of pitfalls that you need to think of when you're designing a system to do this. But if you have this model trained this way, then you can use it for zero-shot predictions. You have some random image, and you have a set of possible text captions. You can just calculate n cosine similarities, take the maximum of that set, and that is the caption that is best associated with this image. And they went on to show that CLIP does very well in this task, and I'll show one plot that sort of demonstrates why CLIP is particularly effective in a bit. But first, I want to talk a little bit more about the sort of mini batch process. So let's say we have a very, very small data set of 4 captions and the related images. During every iteration, what CLIP does is it sort of just randomly samples from the captions and the related images, right? So we're just going to draw 2 out. And the goal of CLIP is to accurately pair the image with the caption, right? We want CLIP to be able to sort of say that the first image in the batch goes with the second caption in the mini batch, and vice versa for the other pair. And then the next iteration, we're just going to do that again. We're going to sample 2 more, right? And we're going to see how well CLIP can do. And this means that how difficult the mini batch is really key to ensuring that the model can train well. And what do I mean by that? Well, this maybe is an easy task, because the image representation for an image of a horse and the image representation for the image of a cat, those are probably going to be relatively, dissimilar. And so it might be pretty easy for the model to recognize that the image of a horse and the image of a cat sort of match up with different text images. If the images were similar, that might be harder. So what would that look like? Well, let's say we draw out both dogs from our set, and now we have to, now the model sees a poodle and a Labrador, and it needs to recognize that that's a harder thing for it to do, maybe more likely for it to mess up. But also that means that there's actually something that you can do to train the model. Like there is a task that the model can fail on so that it can get better. So managing the difficulty of the mini batch is something that we had to think about, and will make a bit more sense when I talk about actually adapting this to proteins. But before I get there, one last point on the image and text clip. One of the reasons why we were so excited about clip and thought it would be a good approach to translate over to proteins is that it's incredibly efficient. So if you compare the green line, that's clip, its performance as you vary the number of images that you train on. And if you compare that to the blue line, which is a transformer language model, which tries to take in an image and then sort of de novo predict or generate a caption, clip is an order of magnitude more efficient. And given that in the protein space we don't have millions or tens of millions of structures, we only have a couple 100,000 in the PDB. This sort of efficiency seems like a very good feature of clip that we could use in the protein space. All right. So the first way that we adapted clip to proteins was as simple as you could be. We just did a sort of control replace. Anytime there was a text reference in the clip implementation, we changed that to a protein sequence. And anytime there was an image in the clip implementation, we changed that to a protein structure. The goal being the same. We want to take a sequence, pass it through a sequence encoder, or a structure, pass it through a structure encoder. And because this is the sequence and structure of GFP, we would want in this learned latent space those representations to be similar so that there would be a high cosine similarity.
Foster, quick question.
Yeah.
Maybe you'll answer this, but how do you represent the structure? Is it by a contact map, or so is it 2D representation? Or?
Yeah, no. So it will be on the next slide.
Okay.
Which I guess I'll just get to now.
Perfect.
So yeah, when we do this sort of control replace thing to translate clip over to proteins, we need to come up with different architectures. We're not going to use GPT-2. We're not going to use like whatever sort of CNET architecture that they used in clip. We use ESM for our text encoder. And for the structure, we use a graph-based message passing neural network. We use Coordinator, which is very similar to Protein MPNN, which maybe is more widely recognized. But both models are based on actually like a 2019 model from John Ingraham at MIT, and it basically takes the backbone of the structure. It translates that backbone into a graph based on nearest neighbor connections in the backbone of the protein, and then it runs some message passing neural net on that graph, and that's how, and that generates per residue representations of the protein, of the backbone of the protein. Yeah, so like this implementation, once we just swapped out those different architectures, we were ready to go train it. One more thing before we talk about training is when OpenAI trained clip, they trained each individual encoder from scratch during the training process. And they could do that because they had tons of GPUs, and they also just wanted, all they cared about was that the latent space was aligned. They didn't care about any individual feature of the encoders. But we looked at ESM, and we said, well, ESM has been trained on millions, tens of millions of protein sequences that our method here is never going to be able to see. Because our method is about aligning sequence and structure representations, we are limited to training on the PDB. So we want to maybe preserve the evolutionary information that Meta AI injected into ESM when they trained it. So instead of training ESM from scratch during this process, we fine-tune ESM. And this is also something that plenty of people have done in the NLP field, like showing that if you fine-tune different parts of the clip process, you end up with different properties. Yeah, so we wanted to fine-tune ESM, and this was work done in collaboration with. Okay, it's good. Dr. Sakshi Jain, who graduated from Alexander Madry's lab at MIT last year. Okay, yeah. So we really. Oh, yeah. Question. Yeah.
What exactly are you fine-tuning ESM to do?
Yeah, so we're fine-tuning on this contrastive task of like we want the ESM embeddings of a protein to match the ESM, the coordinator embedding of the structure. So like, I think I'll have another slide that maybe will make this a bit more clear, but like ESM initially is just trained as a sequence model, right? You mask tokens, and then you generate some ESM representation, representation of the residues, and then the model is trained to like recover the mask tokens. So we now are fine-tuning all of the weights of ESM based on this contrastive task of like finding these correct pairings. Yeah.
The one on the left is like I to IN, and then on the right is like I1 only. Like
yeah. So here I'm sort of showing an example of how this could be used. Like let's say we have some structure, and then we have a bunch of sequences, like the actual sequence of GFP, but also some other protein sequences. In theory, if we've trained this process well, then when you calculate all the cosine similarities of this one structure against all of these possible sequences, the model should recognize that the sequence of GFP is like the correct pairing.
Quick question for me.
Yeah.
Are you fine tuning the MPNN model as well?
Yeah. So
or is that fixed?
That's not fixed. That's actually being trained from scratch.
Oh, it's being trained from scratch here.
Yeah, that's being trained from scratch here. We have experimented. We did a run where we fine-tuned an existing version, and the performance wasn't that different. We think that's because there really are no features of this structure representation that you can't get just by looking at all the PDB basically anyways. Like whereas ESM has this wealth of evolutionary information from the millions of sequences that it's seen that it would never have access to in this training process.
Got it.
Yeah. One question I was wondering, so ESM already has been shown that you can extract structure from it, like like what if you just use that as your matching criteria? How does that compare?
Well, yeah, that is an interesting idea. I mean, one thing is that the contact maps are not that accurate because I'm going to show that we can improve them with this process. I'm also not exactly sure like what you would be matching.
Let's say if you want to know, does this structure match those sequences? You could theoretically pass.
Oh, I see.
Those sequences and get a contact map. And
yeah.
How does that contact map match your?
Yeah, I think that if we were just focused on improving ESM, that would be an interesting approach, right? It would just be like a different task.
Just for the task of matching.
Yeah, but like that's just going to rely on ESM's performance, right? I mean, and I guess what I was thinking is you could fine-tune ESM then to do the matching better, but you would still just really end up with like a better version of ESM. This approach means that like we can just take sort of any given sequence and structure and embed them into the latent space and see how well they match. In a way that I think even if you had a perfect, even if you had a much better version of ESM, I don't think that it would be as good. I think what you're essentially getting at is instead of doing this latent space thing, we could just like predict the structure from the sequence, right, and see if the predicted structure matches the actual structure. And that is what AlphaFold does, and that's what I'm going to compare this method to. And we'll sort of get there when we get there, but yeah, I think that ESM would be just another way to do that with another way to predict the structure, but I just don't think it would be as good or good enough really at all. Yeah.
Thanks.
Okay, so this is what the mini batches would look like in this first implementation of CLIP for proteins, where in a mini batch there are whole proteins, and we can use ESM to generate a representation of the entire sequence, and we can use coordinator to generate a representation of the entire structure, and then we want CLIP to pair up those representations. Yeah, so this is what I was talking about when I mentioned we were fine-tuning ESM, and as Sergey pointed out, one sort of cool feature of ESM is that ESM can be used to predict contacts. We looked at that and said, well, when we do this fine-tuning process, the underlying ESM model is still a sequence-only model. It will only ever take in a sequence and generate a representation. But because of this CLIP fine-tuning process, we were wondering if it now has some sort of indirect structural supervision that's been applied to its weights that might have imbued it with some structural knowledge that it didn't have before. So we wanted to know if the contacts that you get from a CLIP fine-tuned version of ESM would be better than the baseline ESM. That's going to be one of the first benchmarks that we use to see whether or not this training process has worked. It's have we improved the underlying ESM model. Okay, this is what happens when we train the model. The accuracy shoots up incredibly quickly to like 100% in a few iterations, and the validation accuracy goes up to like 80%, and then the model starts overfitting. And this accuracy is the matching accuracy. So 100% matching accuracy means that in our mini batch, the model was able to correctly pair up the structure and the sequence 100% of the time. And we thought, wow, that's pretty high, maybe worryingly high. But let's take a look at the downstream tasks and see if this training process worked. And it did. So yeah.
Because I've seen also in my ML models, like, is it known why validation at some point goes down?
Yeah, that's just sort of standard like overfitting. The model is starting to memorize the training data and not generalizing well to the validation set.
Foster, how in steps like, I guess, around step 25 or where it starts to plateau and you're getting really good accuracy.
Yeah.
How many proteins has the model seen? Has it seen the entire PDB at this point or no?
That's a good question. I would have to double check, especially because this version with, I don't exactly know that number off the top of my head. But I don't actually, I can be pretty confident in saying that it's not the whole PDB yet because we didn't train this model on the full PDB initially. It's like a smaller set. So it could never have seen every structure. But clearly something has sort of gone wrong in this training process because the underlying ESM model has not gotten any better. So here we're looking at the contact prediction accuracy. So we take a pair of residues in the protein in the sequence, and we task the model with doing this binary classification of predicting whether or not these two residues in sequence are close to one another in the structure. And we have two kind of categories, which will make more sense a bit on the next slide. We have close residues. So those are residues between 6 and 24 amino acids in the sequence, and then far residues, which are more than 24 residues away. And on both classes, clip ESM is doing worse than an ESM, meaning this fine-tuning process hasn't worked the way we wanted to. As we were looking into this, and the reason we have these two categories is we noticed that ESM had sort of very divergent underlying performance on these two classes of pairs. So for very close pairs, ESM really likes to predict that those pairs are in contact with one another. And for far apart pairs, it has sort of somewhat of a tendency to think that those don't interact with one another. And particularly the true negative rate, so this bar here is quite poor. So ESM has a very difficult time recognizing that two residues that are close to one another in sequence aren't actually close to one another in structure. So this is something we're going to keep an eye on later.
One question.
Yeah.
So when you say it got worse, is it you trained like a new contact head?
Yeah. So we do have to train a new contact head because the underlying representations have changed, but we don't think that process. I mean, we experimented with retraining a contact head on the old ESM embeddings, and we were able to recover the sort of the normal contact head performance. So we're pretty confident this was not an issue with training the contact head, especially because we were eventually able to get this to improve. So we think this was an issue with like the underlying embeddings weren't getting any better, and so they weren't more informative. Or I really should say the underlying like attention weights, because the contact map prediction comes from the attention weights in the ESM model. But yeah. All right. So how do we fix this? Well, we adapted CLIP instead of taking whole proteins and whole sequences, and instead into a sort of residue level alignment, what we call RLA, of sequence and structure information. So now our sort of mini batch is just a single protein, and because both our structure encoder and our sequence encoder can generate per residue embeddings, the task is now to identify pairs of sequence and structure embeddings on a residue level instead of on a protein level. And that kind of looks like this. So we're still going to have our structure and sequence data sets, but now we're just going to pull out sort of an individual protein, chop up its structure into the individual residues, its sequence into the individual residues, and ask whether RLA can pair those instead of pairing whole sequences and whole structures. And the reason why we think this might help is because of this mini batch difficulty issue that I mentioned before, that we had some sort of memory constraints. We were training on an NVIDIA A100, so a pretty big GPU, but still, because we have to fine tune ESM, and we're using the 150 million parameter version of ESM, and coordinator is a kind of big model as well. We were only able to load like a couple of, well, more than a couple, but only a few proteins into our mini batch. And if there are only a few proteins in your mini batch, and one happens to be pretty different than the rest, then that's going to be the sort of easy task. And we tried some like structural clustering by class, but weren't able to make this whole protein mini batch. We think weren't able to make it difficult enough. But this task is a lot harder because the structures for each residue, they're very close to one another. You can have a sort of repeating structure come up later in the structure. In the sequence, you obviously have the same residue, just at different positions in the sequence. And so this is a harder task. We did also have to make some modifications to the architectures. Both architectures used to have a positional encoding that was part of the featurization, and that would obviously allow the model to cheat in a big way because it would know that this is the first residue in the structure and the first residue in the sequence, and so on. So we had to make sure that the model wasn't cheating in that way, and then we were able to train this residue level alignment process. And when we do this, and then retrain the context, yeah.
Quick question.
Yeah.
About this. So when you're computing the structural embeddings for this case, is it from the full structure where you can compute
Yes.
distances from residues and residues? It's not that you separate.
Yeah, we don't
the
separate it
structure
out.
and then compute the embeddings.
Exactly.
You compute the embeddings and then you just, okay, you have
Yeah,
them
and it's the
from
same
full
thing.
structure.
Right, and it's the same thing for ESM. We embed the entire sequence with ESM,
Right,
and then
right.
yeah.
Okay.
Yeah.
So the original version of the structure embedding had some kind of like global pooling function in order to create like a global representation of the protein.
Yeah, we actually had to sort of build that in to do the first implementation of CLIP.
So now you're not.
Yeah.
Right.
Yeah.
Okay.
All right. Yeah, so this was our first sign that we were doing something right, is we were able to improve the contact map prediction from ESM, and particularly the true negative rate in this close category got a lot better, which gave us some confidence that we were actually sort of imbuing ESM with structured information, which indicated to us that the latent space that we were learning was going to be informative, was going to provide a good scoring function. Okay, so to sort of summarize the method, what we've done is we've developed this residue level alignment, RLA process, which is a contrastive learning approach inspired by OpenAI's CLIP method for combining sequence and for combining text and image information. We trained it using a training objective that maximizes the cosine similarity of true residue pairs relative to mismatched pairs. And our first indication that things were going the right way is that the RLA process improved the underlying ESM model by providing better structural awareness, specifically for this true negative rate. Okay, so that's it for the methods, and we're now on to the results. Okay, so there can be three. Oh yeah, before I get to that, our goal here, just to reorient ourselves again, our goal here is that if we have a set of possible designs, we want to be able to do some RLA calculation that gets a distribution of the designs and then can be used to select the best designs. So all these experiments will be about this general process. So I need to talk a little bit about how we actually get a score from RLA.
There's one online question. If we have sequence but no structure, then can we predict structure and then use the approach?
Yeah, I would say so. I would be a bit careful at what structure prediction method you use. Like, I don't know if you used ESM fold, then you're kind of aligning like one ESM-based representation with something that is fundamentally a different ESM fold representation in a way. So maybe use AlphaFold or use ESM fold and see if it works. We haven't done any work with that yet, although we're kind of getting there. But we've done more work, as you'll see, with what happens if you have structure information but no sequence information. So that'll be coming up. Okay. Right, so getting back to how we actually derive an RLA score, let's say we have some chain C, sequence embeddings, I said U, and structure embeddings V. Well, for a monomer, for a single chain, we're just going to calculate the cosine similarity between the sequence and structure embeddings for every residue and average them, right? So every residue gets included in the score. Things are a little more complicated for multimers, because now we have two chains and we also have an interface, I, and we just sort of empirically noticed that we get better performance if we don't just average over every residue in the complex, but instead if we only average the cosine similarity between the sequence and structure representations of residues at the interface, right? So now we're sort of specifically looking at the interface between these two chains to get the score. Okay, and very related to this question from Zoom, we have a sort of full version of RLA, which is the case when you have the sequence and structure of both binding partners, right? So that's RLA all info. But especially for binder design pipelines, there can be many cases where you have a structure but no sequence, because a lot of design methods produce a backbone first, and then a sequence has to be fit to that backbone. So we wanted to see how well RLA could do in that stage of design, right? Before there's a sequence for the binder. So here there's a sequence and structure for the target and a structure for the binder, but no sequence. And to handle this case, we can just represent the sequence as a series of masked tokens, and then just still plug that into ESM. So functionally, it works quite similarly. Of course, you are missing a lot of information, but the question is, how much does that matter? And I'll leave that for the results coming up. Okay, so at the very beginning, right, I talked about how these computational scores have some efficiency effectiveness trade-off, and I said that one of our big selling points is that RLA is incredibly fast, and here's that plot. So approximate evaluation times of RLA, which is about 1/10 of a second, just applying the Rosetta energy function, so none of the like fancy relaxed stuff, and then AlphaFold 2 initial guess. And RLA is about 100 times faster than AlphaFold 2 initial guess because it's a smaller model. The scoring process is not as complicated. You don't have to do any recyclings, although I'm not actually sure how many recyclings AlphaFold 2 initial guess does. But regardless, you don't need some complex confidence metrics. All you need is the embeddings and then the cosine similarity. So it's a lot faster. All right, and now we're going to go through 3 experiments that demonstrate the effectiveness of the RLA score. We're going to start with monomers, then go to multimers, and then I'm going to end with talking about some very ongoing work on using RLA as a filter, sort of just setting up that problem. All right. So the first experiment, we want to test whether the RLA score identifies compatible sequences and structures for monomers. And we started off very simply by thinking, well, if we have this latent space where there is some alignment that recognizes sequence structure compatibility, then if we mess up that sequence structure compatibility by changing the structure or mutating the sequence, we should see a sort of divergence in that latent space. Right, and we're going to do that sort of by adding some noise to true pairs in the PDB to get from the left space to the right space. And we want to make sure that the latent space looks like it does on the bottom, where it's combined, like they're overlapping on the left and then not overlapping on the right, or less overlapping. Okay, so structure change is really easy to apply. We can just take every coordinate in the structure and just randomly add noise. Okay, and what happens when you do that? Well, on the far right of this plot in light blue is the distribution of RLA scores for a bunch of proteins from the PDB, and their RLA scores, right? On average, about 0.4. RLA scores are essentially a cosine similarity. So a higher RLA score means that the sequence and structure are more compatible. And as you add more and more noise to your structures, you degrade the sequence structure compatibility more and more. The RLA score reflects that. It gets closer and closer to zero, zero cosine similarity, which would indicate no correlation between the sequence and the structure. We've also done something similar with sequences. We can, apply single amino acid mutations to every position in the structure, and we can classify these mutations according to their Blossom 62 scores, whether they're benign, so they're not likely to change the sequence structure compatibility, or they're adverse, meaning they are more likely to change the sequence structure compatibility. If we just look at the RLA ESM embeddings for these mutated residues compared to the wild type, we see that RLA ESM is able to pick up on the difference between an adverse and a benign mutation, because the representation changes more when you make an adverse mutation than when you make a benign mutation. Okay, so that was nice sort of validation that the latent space behaves as we would expect, but we really want to know how well RLA does at this sequence structure compatibility task relative to the state of the art, which in our case is AlphaFold. So we turn to a paper from Sergey's lab where they developed a version of AlphaFold called AF2 rank that, instead of operating as a structure prediction model, instead operates as a structure scoring model that uses the AlphaFold2 confidence metrics to essentially rank a bunch of decoy structures. And here they show that this AlphaFold2 confidence metric correlates very well with the decoy quality. So the decoy TM score represents how similar the decoy structure is to the real structure, where a score of 1 indicates that the structures are identical, and a score of 0 means that they're totally dissimilar. So we took their data set of CASP predictions. They took a bunch of server predictions for proteins in the CASP competition, and they have a bunch of decoy scores, and then they calculate a bunch of AlphaFold scores, and you can sort of plot this distribution of over all these proteins. And generally, AF2 rank does just very good at this task of recognizing when a sequence and structure are compatible. And we just simply ran RLA on all those proteins, and we get a very similar distribution. We perform pretty similar on this task, which is somewhat exciting given that RLA is a lot faster. So even this is a good result for us, considering that we're so much more efficient than AlphaFold. Okay, so the next experiments will be on multimers. We basically want to take the same underlying test of a bunch of decoy structures, and we want to see how well RLA can do at identifying good ones from bad ones. So what does that look like? Well, that means we really have 2 steps to all of these tests that are going to come in this second set of experiments. Step 1 is we want to get some distribution of multiple complexes with some different score assigned to them. And we're going to have 2 types of data sets, one with sort of one more synthetic data sets where the score is a DocQ score. DocQ is like a template modeling score, TM score, and then it indicates how close the predicted structure is to a real structure, and it's used for complexes. A score of 0 is the complex is not similar at all to the real complex, and a score of 1 indicates that it's identical. We also use some experimental binding energies for determining whether a binder is good or bad. But regardless, for whatever score we use it to establish a cutoff, and then we just apply RLA as a binary classifier and calculate some raw curves and some PRC curves, and calculate the area under the curve to get RLA's performance, and we're going to compare that to AlphaFold. So that's going to be the sort of basic setup for all these tests. Okay, so the first test we did was with protein-protein complexes, and we sourced the data of complexes from Capri. So Lessing and Wodak, they took Capri. Capri is a computation like CASP, but for protein complexes. So there's a sort of set of held out protein complexes that crystallographers agree not to release, and then people can make predictions for what those structures are. Some of those predictions are good. Here, they're plotting the percent of models that are acceptable or better. And so some targets are easier than others. But just generally for each target, there are 10 in the data set, and there's tens of thousands of predicted complexes, and some of them are going to be acceptable, and others are not. And the question is, can RLA identify the acceptable ones? Okay, so let's just look at the area under the ROC curve, comparing Rosetta, AlphaFold to initial guess, and RLA. And on this data set, RLA performs a little better than AlphaFold to initial guess. Now, I mentioned there were 2 ways of using RLA. There's the RLA all info, and also RLA without the binder sequence information. So what happens when we ablate that sequence? Question. Yeah.
So for the F2 initial guess, do you have the structure for one of the targets present, or how do you set that up?
Yeah, so AF2 initial guess is actually based on taking the complex structure and providing it as like a template to the model, and then using it to sort of score how well that template, like that design template is, like how good that template is.
I guess for the structures that are in the competition, do you have a template?
Yeah. But because we use the predicted complex structures. Like the whole idea is we're trying to score these predicted complex structures. Yeah. Okay, so when we ablate the binder sequence information, we actually find that it doesn't have that big of an effect on the RLA scores. It's still able to recognize the compatibility of the binder structure with the sequence and structure of the target. And this is cool because this is a task that a structure prediction method like AlphaFold isn't really built to do, because it is built fundamentally to take in a sequence. But we don't need the sequence, at least, of the binding target. Okay. So the the next test here is for protein peptide complexes, and this is done in collaboration with Lindsay, a PhD student in the Keating lab. Yeah.
Topics, so Jerry, I was asking, for your monomer mutation task, did you make single mutations?
Oh,
RLA, understand mutations that are known, disrupt holding or function?
Yeah, so
requirements?
back here.
I think you showed lots of ones, but were there any? No, I think Jerry's trying to know, like, are there known experimental?
Yes, yeah. So we only did single mutations, and we have also used these ESM RLA embeddings to try and predict the effective mutations. So not quite like necessarily affecting structure, but we have data sets of like experimental mutation to some like either stability change or binding energy change. And yeah, we've used RLA ESM and compared that performance to using the baseline ESM embedding, using a couple of other methods. And I don't know if I have that in this, maybe I have it at the end, but the punchline is that we improve the baseline ESM. So this is something that ESM has been known to be able to do. We improve that performance, but to be honest, that performance is not that impressive relative to some other methods that exist, like thermo MPNN or coordinator, actually, in just predicting the energy of a mutation. So yeah, so I hope that answers the question. This is just single mutations, and we have looked at its ability to predict energies. It's better than ESM, but it wasn't good enough to include in the main section of the talk. Okay, so to generate the decoys in this data set for protein peptides, Lindsay used CABSdoc, which is a sort of a stated, well, somewhat state-of-the-art binding mode prediction program. So we used it to do 27 targets from the PDB that are known protein peptide complexes, and then ran CABSdoc simulations to generate 10 overall predictions per target. And as you can see from this CABSdoc plot, each simulation involves running like a lot of possible, exploring a lot of possible binding modes, and there's like a CABSdoc energy function sort of that scores them. And you end up with some predictions that are good, some predictions that are bad, and the RLA task is to identify the good ones from the bad ones. All right, and again, well, here we do sort of significantly better than the existing methods, and that remains true when the peptide sequence is ablated. So these were two, like, yeah.
So for this problem, was there a known true answer for the peptides?
Because we took these known protein peptide complexes from the PDB.
Okay, they were from
and
the
then
PDB.
regenerated predictions using
see.
CABSdoc. Yeah, but there was a bit, a true. I mean, okay. So it'd be difficult to say because maybe some of these CABSdoc predictions could bind in just a different binding mode that's not represented in the PDB. That is a possibility.
And so were there degrees of correctness somehow?
Yes. So we've also looked at that.
How do you assess?
Yeah. So you can use
partial correct answer.
Right. You can use this DocQ score, which, like a template modeling score, will give a value from 0 to 1, where 0, like towards 0 is not as correct, and then towards 1 is more correct. And for to generate this ROC plot, we have some cutoff that's like well defined based on what's an acceptable value. But we've also just looked at the correlation between RLA scores and DocQ scores, and it's better for RLA than for the other methods. Yeah.
So there are many East display data, like in the DR binder design paper, they have like East display with many designs.
Yeah.
Did you test with your algorithm?
Yep. Yeah, that's coming.
Okay.
Yeah. Okay. Right. So both of those data sets were synthetic data, really. And they have some issues, like Nick was pointing out, where we don't really know that the binders that weren't like the native, and maybe those did actually bind and it just not reflected in the PDB. What we really would like is some experimental data that offers better confidence that some binders are good and some binders are bad. So the Keating lab, this is work done by Sebastian Swanson for his PhD thesis, has designed and tested thousands of peptide binders to a bacterial protein called RelE. I'm not going to go into his method, but it involves having some thousands of seeds that are sort of found from the PDB that could potentially interact with this protein RelE, and then stitching them together. And Sebastian tried extending the sort of known binder or generating binders de novo. And then DIA has done a lot of experimental work, doing different display methods to get and screening methods, library screening methods, to get an enrichment ratio that indicates sort of an experimental value of that indicates whether a binder was good or bad. And here, RLA does slightly worse than AlphaFold initial guess. So we do have a couple theories, and I'm going to talk about one of our sort of most supported theories of why the performance on the synthetic data sets that we were using before and the experimental data sets that are going to come after is different. So we'll get to that later. But we were thinking, well, how could we still sort of present RLA in a good light? Like, how could we still make use of RLA in this case, even if its sort of raw performance is slightly worse? And we noticed that, well, RLA is 100 times faster than AlphaFold to initial guess. So what if we developed RLA as a pre-filter, where I'm just having some sort of arbitrary values here. Let's say you have 40,000 designs. You can pass those 40,000 designs through the RLA pre-filter, and you can do that also without a sequence assigned to them, which is something that is less easy to do with AlphaFold. And then let's say you just take the top quartile of RLA scores and then ran your expensive calculations on that top quartile to generate more confident predictions. So we call that our joint method, and the joint method on this data perform much better. All right.
Foster, it's me again.
Yeah.
So in this, if you go back a slide to your one more, yeah, this one. So what I'm thinking is, what is the slow step in design of a binder?
Yeah.
And to me, the slow step is not the design of the sequence, but it's the generation of the structure. And so I'm just curious what you're thinking here. If you still have to generate 40,000 different structures, then the bottleneck to me would be the generation of the structures and not necessarily the design of the sequence. But is it that you can, usually with protein design, we try to assess a structure based on, for example, designing some sequences and then folding them with AlphaFold.
Yeah.
Is the compelling, whittling down argument here that you could avoid that part, but it's still slow to generate all the structures?
Yeah. So it is still slow, although there are faster and faster methods being developed every year. Like diffusion methods are a pretty fast way to generate a lot of designs. But separate from that, like a selling point is just that RLA is so much faster that you could run this on hundreds of thousands of designs. Instead of having to run AlphaFold on your entire set, however many designs you've generated, however slowly that process was, it's still going to be a lot slower to have to run AlphaFold on all of your designs as opposed to running RLA. And if RLA is effective enough, then the efficiency gain that you can get from that could sort of tip the scales towards using RLA as a pre-filter. But I mean, it is a good point that this is faster than AlphaFold 2 on scoring, but is scoring the slow step? That's a question that we've gotten. Absolutely. Okay. So now we're going to do some more of this same style of test with mini protein complexes. This is going to be that Baker Lab data they generated, paper Cow et al., where they generated like tens of thousands of different designs for a bunch of different targets, and then experimentally validated which designs worked, which designs didn't. And this plot is from the AlphaFold initial guess paper, where they showed that using these AlphaFold 2 confidence metrics was the best way to filter designs. This is still a very hard task. Even AlphaFold, the best performance it gets here is 7%, meaning that 7% of the designs that were past the AlphaFold filter actually worked, which could be a lot higher. So when we evaluated RLA on this task, similar to the RLE data, but to an even bigger extent, like RLA wasn't working as well, and only 2 of the proteins did RLA outperform AlphaFold. We also here, I'm going to show the area under the receiver operator curve and the area under the PRC curve, because there are so few successful binders. Often we have a very imbalanced data set. It's helpful to look at the area under the PRC curve as well as the area under the ROC curve. But I mean, it still demonstrates very clearly here that like RLA not performing that well. But the join method does a lot better. It's less clear to see in the area under the receiver operator curve, but in the area under the PRC curve, it is sort of quite clear that you get the best performance with the joint method. Not only is the joint method faster because you don't have to run AlphaFold on all of your structures, but it is also a more effective filter, a more effective scoring function. Okay, so yeah, I think before I show that result, I mentioned a little bit ago that we had this a couple of theories as to why RLA's performance on the experimental data sets were different than the performance on the synthetic data sets that we were using. And the hypothesis that I'm going to sort of show is about sequence variation in the data set. For the synthetic data sets, there was only one binder sequence, and there were just a bunch of different binder structures that were either generated in Capri server predictions or generated by Kabslock, but there was only one binder sequence. For all the experimental data, there is one binder sequence per design, because in order to get that experimental data, we had to take the structures, fit a sequence to them, and then actually make them. Or well, Dia did, and the Baker lab had to. I don't touch the experiments. So we were thinking, well, maybe because we also know that RLA can do pretty well without the binder sequence information. So maybe it's just more sensitive to structural changes than to sequence changes. And if you have a data set in which there could be multiple structures that look very similar, but one of them binds, one of them doesn't bind based on the sequence, maybe that's kind of what is throwing RLA off. And so we wanted to take these experimental data sets that have sequence variation and desequence them. And I'm not going to talk about how we did that, but it essentially involves some structural clustering and then defining success for these structural clusters based on the success of the members that comprise them. But what's important is that when we do this structural clustering process to desequence the data sets, the performance of RLA improves significantly on both data sets. So maybe this is again pointing to using RLA as this very early pre-filter where you don't have any sequence variation, you just have structure variation, and RLA there could be a very useful tool. All right, so the last experiment, really I'm just not going to show any sort of results here, just going to set up the problem and how we're thinking about going about it. And this is work that's being sort of pioneered by Jesse, a undergrad summer student in the lab. And the idea is to take our target, in this case, again, REL-E, and to use RF diffusion to generate a set of thousands of different designs, and then apply some different filters. So we have RLA as a filter. We've also been working on developing this very simple, like a number of contacts filter that is also very cheap and easy to calculate, just like RLA is, but and also just structure based. And then the idea is to apply this filtering process, run the different filter designs through the expensive scoring method like AlphaFold to initial guess, and see how many hits you get when you apply these different filters. And of course, we're maybe eventually with some new postdocs that are joining the lab as well, going to be experimentally testing some of these designs down the road. So that is very much the future of using RLA is in some way as a generative process. Yeah, a couple other things we could talk about in the Q&A sessions of interesting applications of RLA as well. But I think now it's time to wrap up. So as a summary, what I've talked about is developing this RLA process that aligns sequence and structure embeddings in a shared latent space. I showed that RLA supervision process improves the sequence encoder, so you get this fine-tuned RLA ESM model that is better than the initial ESM model at predicting contacts. The latent space can be used to assess sequence structure compatibility in monomers and multimers. And I briefly mentioned how you could potentially use RLA in a binder design pipeline. So some acknowledgements, I think I've sort of shown some of these names already, but the whole Keating lab, and especially these people that I mentioned throughout the talk, and Swati Jain from the Madry lab, and my committee members, Professor Bruce Tudor and Joey Davis. And yeah, I'm happy to take any questions.
Nice talk, thank you. Do you think your model performs better for the test cases because they are more natural sequences, so ESM has seen them, or like it's trained on natural sequences, and all the de novo sequences are just further away from what ESM has seen, and so it's like has a harder time predicting them?
Yeah, that is interesting. I mean, I know that like maybe the people at Meta AI or now the new company they have would say that ESM is like just as good on de novo sequences as natural sequences. Or maybe they'd hope that's the case, but I don't know if they've really looked at that. Yeah, you can maybe look at just how well ESM can do or RLA ESM can do on the initial ESM task of mask token recovery, on de novo versus natural proteins. I don't know if anyone's done that, but that would be interesting. That's a good hypothesis. Yeah.
Very nice talk. I have a question about the combined. So you showed that by combining RLA with AlphaFold, you achieve a better accuracy. So I was wondering, how do you interpret that result? So does that mean that the two methods are filtering out different proteins? Have you compared like what kind of proteins it filtered out?
Yeah, that's exactly it. It turns out that the AlphaFold score and the RLA scores are very orthogonal. So we think that the RLA process kind of filters out some things that AlphaFold is not as good as filtering out, and that then when you run AlphaFold on that filtered set, you're sort of biased towards getting a better performance. Yeah.
Hello. Oh, cool. So this is a question back very early on when you were talking about improving the cosine scoring of things. What's the vector space that those objects are living in?
Yeah, so for us, it's R320. Okay. I don't know what it was for CLIP, probably maybe a lot bigger than that. But for us, yeah, R320.
So is the process to improve the cosine scoring some sort of basis vector rotation?
No, well, the model kind of figures that out, I guess, right? Like we're not directly formulating some process to change the representation. We're just asking the model weights to produce representations that have a higher cosine similarity. And we're letting the model weights figure out how to do that.
Okay, thank you.
Maybe had a question first. Hi, so I was wondering, it's interesting that the model works so well when one side is masked, right? One question I had about that is, have you tried, maybe for those problems where you're using that score as benchmark, when you feed it decoy sequences, I guess where does the all masked sequence lie in that sort of, score landscape, right? Like presumably if you fed it the right sequence, it would get the best score and there's some minimum score. Like why does the mask, like is it just sort of in the middle? And so you're,
I think I forget exactly what we've done here, but I think I've tried instead of replacing the sequence with like X tokens, which for ESM is their mask token, I've tried replacing like glycines just to see how well that works. And I think that works worse. Like there is something about giving ESM a mask token that it can say, okay, like I'm not going to trust any of this information. Like I'm going to instead let the other sequence information that I do have from the target and predominate. Maybe we need to do a little more work to really show this, but yeah, I wonder if when I give it the glycine sequence, it thinks that there's a bunch of glycines there, and that's like not the case.
Great talk. Yeah, I have a question about when you mentioned about this peptide binders generated by the diffusion, and then you filter apply a few filters. What did it look like after you used ROA filter to these binders?
What did it look like in terms of like, were there patterns to what structures were eliminated? Is that your question?
Right. I guess how long are these binders and what?
Yeah, I mean, so they really vary. I mean, the mini protein designs from the Baker Lab that we tested, I forget the exact number, but I think they're probably like around 100 amino acids in that ballpark. But the peptides that we tested were more like 30 to 40 to 50 amino acids. So we have tested on data sets with various sequence lengths. Yeah, and we don't think that RLA is. We've made sure that like RLA is not just picking up on sequence length. Like we've sort of normalized to the length of this sequence when we're calculating the score and things like that. So.
Right. Does the RLA improve the binding, I guess?
Well, I guess we've sort of shown that it's a pretty good scoring function, right? We are looking a little bit into like actually using RLA as a generative process as well as a scoring process, which is maybe what your question gets at. You could think of this, if this is a good scoring function, then it's pretty natural to incorporate it into a generative process where as you're doing some design, you want to include this RLA score as like an objective function. But we haven't really sort of started doing that yet.
Thank you.
While I'm walking down, related to that last question, is there a bias at all to what the model thinks is a favorable binder tertiary structure? Is there, like RF diffusion probably has some bias to generating helical proteins, but is there some bias to your model?
None that I've, I guess, found. But I haven't done a sort of very systematic search of different like tertiary motifs. But I have, so in the Baker lab, especially when they were generating their designs were categorized into different structures, and those different structures have different alpha helical, and or beta strand propensity. And I didn't notice any sort of bias towards a specific type of design. So to the extent I've looked at that, no, but I don't really know the answer to that.
So one of the cool things about the clip, as you talked about, is that it's very data efficient, right? And something that we think about a lot in Nick's lab, right, is sort of protein ligand interactions, and it's also something that's very data limited. And I'm wondering if you've thought about, or if you see sort of a natural extension of this method where there's sort of an additional clip training process to align a like chemical fingerprint embedding with the ESM embedding before sort of repeating your RLA process with like an all atom diffusion model or something in order to like fully understand molecular information and maybe take advantage of the fact that amino acid letters are still like chemical fragments.
Yeah, so I thought about that a little bit. There is one sort of general issue, which is that let's say you wanted to improve. One good thing with CLIP or the RLA process is that it's very modular. Like if you come up with a better sequence encoder architecture, just swap that in for ESM and like great, the model should do better. So I think what you're kind of getting at is instead of having a structure representation that's just based on this graph neural network of the backbone, maybe the structure representation could be somehow like all atom. Yeah, I have thought about that. The one issue is that if you really did give an all atom representation of the structure, I think the model would be able to cheat really easily because it knows then if there are more, like certain amino acids have a certain number of atoms in their side chains. So maybe then you wouldn't want to get, so then, yeah, like what's the benefit to doing an all atom representation if the model is going to cheat in that way? Sort of unclear. And then the small molecule question or like, fingerprint, then I'm not sure what you would do for the sequence embedding because you do need to represent both sides. I'm sure there probably are some, but yeah.
I guess like specifically what I'm proposing here, I guess, is an additional clip that like aligns the sequence encoder with another model that's like embedding the ligand fingerprint or something like that.
Yeah, okay, that'd be interesting. I'd be happy to chat after. Yeah.
Well, hi, Foster. That was a very unique talk and topic. I'm curious about the slide where you showed when you're evaluating the early slides, you added noise to the structure, and it was, I think, like 0.12 angstrom noise.
Yeah.
We know that like protein MPNN has added noise before, and so to me, I guess they claim that it added robustness. And I guess for you, you're saying that adding some noise, it decreases in the quality of the predictions. So
yeah, well, so I think it just depends on when you added the noise. I think maybe we actually should add noise during training so that the model recognizes that structures aren't totally fixed and it doesn't like really overfit to the like exact structure values that are in the PDB. And that's what they did in protein MPNN and showed that it improved performance. But because we've trained RLA just on the PDB, as soon as you mess up that alignment, the scores decrease. But I do think that's a valid point. That maybe we should think about adding noise during the training process.
Sergey, do we have any questions on Zoom?
MS Sullivan.
Okay, well then, everybody, let's thank Foster again for a great talk. Thanks, Foster. All right, and you can pepper him with questions as much as you want now. All right, thanks, Foster. We have somebody lined up. Yes, I should have probably drawn. Oh. So next month we have our speaker is Joanna Slusky for next month. She's faculty at University of Kansas. Come next month; it'll be a great talk. At MIT.
It's really interesting. Yeah. Two questions. First is how much faster is the